presentation is of Dr. Stefano Salmaso. He is assistant professor at the Faculty of Pharmacy of University of Padova. During his career, he completed his training in international research group as University of Paris Sud, Strasbourg, and Boston. His research activities are focused on conjugation of proteins, peptides, and small drug molecules polymers, aiming to improve their physicochemical, pharmaceutical, and biological properties, development of micro and nanoparticles for the prolonged and controlled release drugs, and the development of environmental responsive colloidal system for the prolonged release of drugs. The talk of today is targeting the brain through the ascorbate transporter.
Thank you very much for the introduction, and I want to take this opportunity to thank the organizing committee for this nice event. I had already the opportunity to thank personally Giovanni Tosi, but also the other people that participate in this organization, and which was really stimulating from the scientific point of view and also for the extra congressional events. The talk that I'm going to give in the next few minutes is concerned the targeting of the brain using the ascorbate as the gate of entrance, and it emerged from a collaboration between the University of Padua and Northeastern University in Boston with Professor Torcillin, who probably you are familiar with. Basically, it was inspired because I simply stepped into a publication was saying that there are different biodistribution of vitamins in our body, and one of the vitamins that are most studied is the ascorbic acid, vitamin C, and I was interested in the fact that if you look at the biodistribution of vitamin C in the body, you can see that most of it is present in the brain, not only in the brain, unfortunately, but the concentration inside some neurons can be up to 100 or 200 times than what you can find in the blood. So the question was very simple: why can't we use ascorbic acid as a targeting agent to penetrate the brain and then deliver some drugs, considering the techniques that we can use with the microparticulate system or supramolecular system. Just studying this topic, I discovered that ascorbic acid is taken up specifically by a sodium-dependent transporter, which is expressed basically in the choroid plexus. So we have some specific transporter and the gate to enter the brain. I'm not a physician, so I won't go too much into details about which is the physiology of the gate that we are interested in, but if you open an anatomic book, you will see that the choroid plexus has a very peculiar physiological structure, and what is very interesting is that the capillary at the choroid plexus is a little bit leakier if you compare with the BBB capillaries, and so we can imagine, this is still to understand deeply, but we were discussing with many other people here in this occasion that you can have a passage from the blood to the epithelial or the choroid plexus, even of molecules that are a little bit bigger than water or sugar or glucose, for example. So it is still to be cleared which is the exact dimension that we can filter out from the blood, and then at the end you will have anyway a sort of very tight barrier, which is the epithelial, and these cells basically are characterized by tight junction. So In our idea, I think that this gate can represent a good windows to let a microparticulate or nanoparticulate system to pass from the blood into the brain. So basically what we did was, first of all, to select a good cell model in order to study and characterize any targeted delivery system which can be decorated on the surface with ascorbate, with ascorbic acid. Then we prepare some nanocarrier. We focused basically on liposome, micelles, and gold nanoparticles because they are very well known and they have specific properties that can be exploited for this purpose. We investigate the in vivo specific targeting of this nanocarrier, and then we shifted to the in vivo biodistribution, and we got lately some good results that I will show you throughout the presentation. Basically, we were interested in selecting some immortalized cell lines in order to see which of them were able to express the ascorbic acid transporter. We studied about 10 different cell lines coming from mouse, rat, and human origin. And what we found by FACS analysis is that basically C6 and F98 cells, both coming from rats, they express this transporter for ascorbic acid. You can also find it on 3T3 NIH cells from mouse. They express a little bit less than the other two. And this is also interesting because these two cell lines are at the same time gliomas tumors. So we can have like a double targeting of the system once it reaches the choroid plexus, and if we are able to make it pass through the barrier, then you can have also targeting on the glioma, which is one of the reasons that we are interested in brain targeting. Then we synthesize with a simple method this targeting moiety, which was obtained basically by conjugation of a derivative of ascorbic acid with the commercial available phospholipid PEG. We obtained this targeting unit, we purified it, and then we characterized by a bunch of different spectroscopic systems. We also, of course, at this point, we prepared the liposome with the targeting agent that I showed you before in the previous slide, and also micelles by a simple technique, the thin layer rehydration technique. We characterized both the dimension of the targeted and untargeted liposome and micelles, and what we found is that in the case of PEG liposome, basically the dimension is around 180 nanometers. In the case of micelles, of course, they are much more smaller, and the dimension is just a little bit shifted in the case of the ascorbate PEG phospholipid micelles. The zeta potential basically is a little bit negative, which is of course clear and is due to the presence of ascorbic acid, which is of course an acid, so it tends to deprotonate. We then studied this two system with the selected cell line that expressed the ascorbic acid. In this case, I just show you the result that we got with the C6 glioma cells, and as you can see, basically both with the liposome and micelle, you have an increase in the targeting, which is specific if you compare to plain liposome or PEG PEG-P micelles. Apparently the liposome are working a little bit better because basically they have a different amount of labeling agent if you compare with the liposomes. So of course also the fluorescence that you can get from the cells is a little bit different. We also took some good pictures from the C6 cells, which were incubated with the liposome and also with micelles in the next one. What you can see basically is that in this case, you have an accumulation of liposome inside the cells. We still don't know actually if the fluorescence that you see in the picture is just on the surface or inside the cells, because these pictures are not from confocal microscopy. It's just simple fluorescent microscopy, so we don't know exactly if you can have also a transport of the whole liposome inside the cells. And in this case, of course, these are the cells treated with the PEG liposome, and as you can see, you don't have any red spot inside the cells. This means that, of course, the targeting agent is working fine with this cell, and the same result we got with the micelles targeted by the ascorbate unit. And the first line is about the control that we used, so normal PEG-P micelles. We also demonstrated that the targeting is basically specific. And if you look at the first picture, you see the same liposomal formulation incubated with the cells. And with the C6 cells, you see a lot of red spot in the cells. These actually have different magnification compared to the previous one. We then incubate the same formulation with cells in the presence of excess of ascorbic acid. In this case, the ascorbic acid is just binding the transporter, so you don't have any binding of the liposome that you incubate, and you have a drop basically of the fluorescence of about 5-fold. So basically the technique is very specific for the ascorbate transporter. We then skipped with the in vivo test, and what we did was basically to administer the liposomal formulation to mice through intravenous injection. On the left panel you see the biodistribution in different organs for liposome PEG, and in the right one the targeted liposomes. There are not that much difference. Basically, most of it goes directly in the liver. And then, if you focus a little bit more on the brain distribution, what we were able to see is that basically there is no preferential accumulation in the brain when you consider the control and the targeted liposome. You have as the time passes, you have a decrease in the amount that you can find. I do believe that you can have basically a sort of absorption on the vessels, but this doesn't mean that the liposome penetrates the brain. So at this time the question was very simple. Either we are working with the wrong model with the in vivo study, which also might be the case since the physiology of the ascorbate acid in the rodents is a little bit different than humans. But at this kind we have to change the model and select maybe a different animal. Or another chance is that maybe morphology, surface properties, and also size of the nanocarrier that we are investigating can be changed if they really matter on the results that we got. So we started with this second option, and we tried with the downsizing basically of the carrier that we are investigating. These are really latest results that we got in the last 2 weeks, so they are still preliminary, and we are going to investigate a little bit more about also the chemistry of this nanomaterial and the way we modify the surface, because as you can imagine, they have a very huge surface energy, so they are not that much stable, and we are still working on it. In any case, just for the preliminary result, we did more or less the same strategy in order to modify the surface of the gold nanoparticles with ascorbate linked on a very short alkyl chains, and we modified the 2 nanometer size gold nanoparticles. Then we inject it into the mice, and we check again the biodistribution. Again, if you consider the distribution in different organs, you can't tell that much about the biodistribution. Again, most of the materials just goes to the liver, regardless it is targeted or untargeted. As reference, we produced the gold nanoparticles with the carboxylic acid at the end. But in this case, we got a very nice surprise, because if you look at the control gold nanoparticles, you don't see basically any accumulation in the brain, and basically the profile is very similar to what we found with the liposome injection, and you can see a decrease of the amount of gold that you can find inside the brain. If you look at the targeted gold nanoparticle, you can see that as the time passes, you have a sort of increase, a neat increase of the gold that is accumulating inside the brain. So I think there are good perspective for the future, and we are going to investigate this, and we are very open to collaboration, so we met a lot of people that can help with the brain model, and also with this specific endothelial colloidal plexus barrier. The last slide is about the fact that we also evaluate if this material can be eliminated from the body when the time passes, and the answer is yes, because we found gold nanoparticles in the urines basically, and you can see that about 3% of the total dose is found only after 24 hours. So basically, I think it's very important also to consider the fact that you don't have any accumulation inside the body, because this is usually asked by the agencies that then release the use of this material for in vivo study. The conclusion is that basically you can exploit the ascorbate transporter for brain delivery. The study is very new, so it needs to be investigated in a deeper way, and a lot of mechanistic and way of action needs to be well understood. Basically, beside the specific targeting of the brain, what matters is also the morphology, the dimension of the things that we want to administer. So the nanocarrier is very important, and also its properties concerning the surface and the dimension. If you want to read a little bit more details, you can find the whole part concerning the in vitro study, the chemical part, and the in vitro study is going to be published on bioconjugate chemistry, and we got accepted the paper last Monday, so this is also a time for enjoy the good result we got. And thank you for your attention.
Thank you very much, Stefano, for your nice presentation, as always. So the paper is open for discussion. Giovanni?
Thank you very much, Stefano. Thank you for being here and great work. So and now I'd like to ask you a comment about the use of PEG, because I read in literature 3 or 4 articles dealing with an accelerated blood clearance after multiple injection of PEGylated carriers. So I mean, there is a possibility to develop an immune response after multiple injections. So I would like to ask you a comment, because I know you're working with PEG, so it is open to all the audience, I think.
Yeah, okay. This is a nice question, because there was like a discussion table in the last CRS in Copenhagen in July, and basically the audience was shifted between people that do believe that PEG is safe and you don't have any kind of immune response, and some other that have data about antibodies that were found in the blood. So I think that the discussion is still open, and it might depend also in which way the PEG is attached and to which molecule is attached. I think it's much easier to have antibody for PEG when it is attached to proteins, for example, because the structure is a little bit more rigid, and then you have the PEG with the surrounding amino acid. In the case of liposome, as you saw, we didn't got that much results, so I'm not sure we can keep up with the evolution of this system for in vivo study. And as our colleague before showed, there is only one company that is formulating glutathione linked on the surface of liposome. They don't have that much result in vivo, so I think we have to take care about formulation with micelles and liposome if we want really brain targeting, because also with the material that we are investigating, if you consider that the ascorbate transporter is a transporter, so it tends to transport only small molecule, which is ascorbic acid. I believe that this is better than nothing, in the sense that if you really have a binding on that area of the choroid plexus, and then you can also have a release because you reach at least the site of action where the drug is expected. And I wouldn't exclude the fact that you can have a transcytosis once you have binding on this transporter. So there is plenty of transcytosis also of albumin that was simply alkylated and aminated basically. So why not gold nanoparticles that are as big as albumin, for example? 2 nanometer is very small.
Stefano. Okay. Stefano, I have just a curiosity concerning just the rationale, just to choose, of the targeting agent concerning ascorbic acid. Because if I got well, you have decided just to use ascorbic acid because some neurons have an accumulation of this particular compound. Mm-hmm. Okay, I got well.
Well, the very original idea was to use ascorbic acid because there is this transporter in the choroid plexus. In the meanwhile, when we were investigating this process, we stepped into some glioma cells that express the transporter. So we just screened a lot of cell lines and basically choosing among the ones that are present in the central nervous system. So some gliomas, neuroblastomas, and many others. And just by chance we found that some gliomas also expressed this transporter. So concerning the mechanism of action, at the very beginning we were expecting that this transporter is working on the choroid plexus when it is in the physiological state. But then we discovered that the transporter is also expressing some tumors inside the brain. So I think that if it is not working well on the choroid plexus, when you have tumor, you have also the BBB that is a little bit damaged, and you have much higher leakage. So these systems that are very small, they can pass through the barrier and then target the gliomas. So we are still investigating. That's why I started with healthy mice, just to see what happens when everything is fine inside the body, and then maybe we will shift to a model that with the gliomas maybe.
Yeah, I just put this question because, I mean, the rationale of your work is really nice. But probably, just looking at your result concerning the biodistribution in the brain, probably you have a competition because the same receptor, the same transporter is present also in other part of the body, just like liver. And so maybe your, let me say, bad result just concerning the biodistribution, it seems that you have more or less really a few localization, the level of the brain, is not because it doesn't work, but because of the presence of these kind of transporters at level of other organs, just like liver. In fact, we have a really a huge accumulation in liver, especially in the first hours of your experiment. And so it's a nice idea, but maybe it should be better just to change, let me say, the molecule, the targeting molecule. I don't know, just an hypothesis.
Yeah, I agree with you that basically what you find in the body is that this transporter is expressed in some other body district. You can find it in liver and lungs, for example. We are investigating it. We want to understand if it is working in the brain, because I do believe that basically if you can find a huge amount of ascorbate inside the brain, there are some preferential transporter that maybe allow it to penetrate much better in the brain than the liver. And then about the liver, I would also consider the fact that this is happening for any kind of nanoparticulate system, regardless the targeting that you have. So I had the same accumulation of liposome, for example, untargeted compared to the targeted one to the liver. So this means that it is not depending on the targeting agent by itself. It's something that is part of the physiology. But I think that it's also a good platform if we want to investigate some specific targeting to the brain, because I think that this choroid plexus is offering us a lot of opportunity for selective targeting. There are many other small molecules that are transported specifically through the choroid plexus compared to other, like glucose, that is basically everywhere, so you can target it by itself.
So thank you very much, Stefano.
Thank you.